All right, now I'll just introduce our next speaker. This speaking next is going to be Professor Pamela McCombe, who is the Professor and Head of the Royal Brisbane Clinical School in the Faculty of Medicine and co-head of the Brain and Mental Health theme at the UQ Centre for Clinical Research. Professor McCombe is a neurologist who has extensive research experience in neuroimmunology as an NHMRC Senior Research Fellow, and more recently has resumed clinical practice as a neurologist, continuing her research as a NHMRC Practitioner Fellow. Her research interests range from biomarkers and heterogeneity in amyotrophic lateral sclerosis, inflammatory neuropathies, immunology in stroke, pregnancy and MS, and neuromuscular disease. Excellent, and I'll hand over to you, Professor McCombe.
Good morning, everybody. Can I just check you can hear me? Yes. So I'm very pleased to be asked to speak to you. And I have to say, I was thrilled just to hear the preceding talk. Congratulations. I thought that was just brilliant talk. Now, I've been asked to talk to you on this topic, international motor neurone disease genetic databases. And you'll see the way I'm going to tell the story is how I got into the position of being involved in that. Clearly, my role in these databases, I have to say, is as a clinician. So just about myself, it's already been mentioned. I am a neurologist. I have been doing it for a very long time. I'm getting old. I've always been keen on research from student days, and my BSc Med was in molecular biology in the 1970s, but it was so long ago we didn't even have PCR at that stage. I became a neurologist, have a PhD in peripheral neuropathy, and as mentioned, my career's largely been in neuroimmunology and neuromuscular disease, which includes motor neurone disease. Now, genetics is probably the next most exciting clinical specialty for young neurologists to get into, because we're just starting to get clinical trials about actually treating people with different genetic variants. It's going to be very exciting. We're going to end up needing clinicians who can treat patients. Neurology is very rich in inherited diseases, and most of them we know about are Mendelian inheritance. And, you know, it ranges through all the neuropathies, muscular dystrophies, spinocerebellar disease, channelopathies, dementia. So many genetic diseases. And many were first described by the really excellent clinicians of the late 19th century. Names like the great Charcot, Burry-Tooth, all of those were neurologists who clinically recognised and phenotyped people with these diseases. And probably that's what clinicians are really still going to be doing going forward. Interestingly, the first gene to be mapped to a chromosome was in a neurological disease, Huntington's disease, in '83. And it was the first gene to be identified in 1993 through a fabulous triumphant collaboration. And it was a triplet repeat disease, and triplet repeat diseases are very common in neurology. So the way I'm going to tell the story is that, yes, I now participate and collaborate with these large diseases. But of course, like most people wanting to do research, I wanted to have my own lab and do things and be in charge and do it in my own way. But of course, that's no longer really possible. So starting back in the 1980s, we've increased in our ability to test for genetic disorders. Some of the early stuff was separating the alpha-1 antitrypsin alleles by size of protein on a gel. HLA was very big in neurology, as in other diseases in the 80s and 90s, and originally it was done by old-fashioned serotyping, but later we've been able in our lab to use sequence-specific primers. We've looked very carefully at gender HLA associations. Another gene that we studied was called KIR, which is a very pleomorphic gene involved in T-cell cytotoxicity. We've been able to do a GWAS study ourselves by getting the chips and being able to do our own analysis. And we can do small-scale sequencing of this gene we're particularly interested in, which is the promoter region for the NF-kappa B IA gene. And NF-kappa B, as you all know, is a very important cell signaling molecule in many diseases. But of course, these were all small projects that we were able to do ourselves, but they don't really cut it anymore. And now it's necessary to be involved in international teams so that you can have large numbers to have the computational and statistical power. But before I just move on to that, I also want to just tell you about something else, that many of these tests no longer need a research laboratory. And as clinicians, we can directly order many tests and identify many patients. So the triplet repeat diseases that I mentioned are very readily available. The local laboratories can do them. You don't need a special genetic sequencing laboratory, and these are all NATA accredited. Previously, we had to have a candidate gene, decide that we think such and such a gene was the candidate and get the gene sequenced. And such sequencing was not always accredited by NATA, which is the accreditor of pathology labs. But over the last 15 years, there has been chip technology with panels of genes, so you don't have to be quite as accurate with your clinical diagnosis. And this allows easy screening for known genes associated with classes of disease. So where does a clinician's research kind of come in? So what I want to just show you is one of the ways that clinical research can go. And this is actually muscle disease. I haven't got to motor neurone disease yet. But we are able to send out in a clinical kind of a test and find out the variant of people with various inherited muscle diseases. These are myopathies, muscular dystrophies, channelopathies. And what I've done is I've just plotted here the frequency of various different genes. And you can see there are 3 that are more common, which facioscapulohumeral muscular dystrophy, the dystrophinopathies, that's Duchenne's and Becker's, and this one called dystrophia myotonica. So we can send forth and get the information. And what are we going to do about it? So we're currently organising a genetic register for all of Queensland because we know very little about the prevalence or indeed the natural history of these diseases. And furthermore, we will have a list of fully genotyped subjects ready to go when we get more of the clinical trials. I just, you probably go to the butcher's and you buy a steak and you think muscle looks, fairly uniform, but it's actually incredibly complex. And when you think about what its job is, it has to change energy into movement. And there are many, many complicated proteins inside muscle, and these are some of them. And just as another plug for muscle, it's got the 3 largest human proteins. These are giant proteins called titin, obscurin, and dystrophin. So this is not what I was asked to talk about, but this is one way that clinical genetic neurologists will be able to learn more about, as I said, prevalence, natural history, and treat patients. Now I'm going to turn to motor neurone disease. So motor neurone disease, you all know of someone who's had motor neurone disease. It's a horrible disease. It's a severe progressive neurodegenerative disease leading to death from respiratory failure. And over here on the right-hand side, this is our local data. This is the survival curve for 726 patients we've looked after. 50% of people die in 1 year, majority die by, 50% die by about 1,000 days. Most have died by 2,000 days. And then a number of people live for a bit longer. But it's a terrible, terrible disease. It's scientifically interesting because there are many proposed pathogenic mechanisms, including particularly glutamate excitotoxicity. So glutamate is a neurotransmitter that causes toxicity, and a number of other mechanisms, failure of autophagy, dysfunction of mitochondria, and in particular aggregates of insoluble protein. And all of those things are important because we've got genetic variants in many of these pathways. And fascinatingly, for the infectious disease people around, it seems probable that these insoluble proteins actually spread by prion-like mechanisms. Now, we've always known that some patients with MND have apparent Mendelian inheritance. For many of them, the penetrance is variable, but that's probably because you don't get motor neuron disease until usually, well, the peak incidence in our study is 65 years. So if you've died of something else before then, you won't be an informative person. But so there are genes and there is Mendelian inheritance. And as I've just said, the genes that have been implicated include genes involved in these pathways to do with pathogenesis. So this is a slide from this paper, and versions of this slide come out every year, and it shows how things have evolved. So they've tried to show you the different techniques. So originally it was sequencing of candidate genes. Then we moved into larger scale studies until we started to get the GWAS. And now we're at a stage of trying to get more out of GWAS than just the association with the common variant SNPs. But there are various mechanisms to try to find the rare variants also that might be associated with disease. The very first gene we had was SOD1, and then that was back in 1994. And then these are the different genes. But this author has tried to show that they're not all equally well proven to be causative genes. So the green ones are fine. They've got ample replication data and good evidence of pathogenicity. The yellow ones are not quite so good. And the red ones are in the list because they may well pan out to be important, but we really shouldn't count them as being causative at the moment. And then another issue in MND clinically is the patients don't all look the same. There's wide heterogeneity, and there's also overlap, genetic overlap. Some of the genes sometimes cause motor neuron disease, but sometimes they cause something slightly different. So these are the genes that have been discovered. And this is where we're going with the large scale collaborations. So we first got involved with the research that led up to our involvement in the large consortium by trying to get sequencing of our patients. This is before we had the chips, before we had any easy way of testing. And we've had a major collaboration with the people over at IMD, particularly Naomi Ray and Fleur Garten. And the first thing we wanted was could they sequence our patients and tell us what genes they had? So in fact, we did this. It was done a fair time ago now, finally published in 27. And we did whole exome sequencing of 120 patients. And we didn't get a real lot out of it, as you can see. We found 4 known variants and 5 variants in genes that were neurogenes. But then we discovered that exome sequencing wasn't going to find all our genes. And there is one of these repeat expansion diseases called C9orf72, which is a fascinating story. The historical geneticists have shown this probably arose from a single mutation, someone up in Scandinavia about 400 years ago. And now it's given the world motor neuron disease and frontotemporal dementia. But we couldn't get that gene out of the exome sequencing, so we had to make other arrangements. So now we've moved on, and we now can test in what I mentioned, a more clinical situation for MND genes at our clinic. And this is looking for the Mendelian inherited genes. And this is what we're up to at the moment. We've tested nearly 300 patients. We haven't tested all 800 of them because we're still getting used to the genetic counselling required for people who are not familial. But this is what we find. The majority of our patients have that C9orf72 gene. The next biggest one is the SOD1 gene, the first gene ever found. And this is what we find in the other patients. All right. So now we're getting closer to the consortium. So what about the people with what we call non-familial ALS? We used to call it sporadic ALS, but we're not at all confident that these patients are truly sporadic. If we hunt back far enough, we can find what's called cryptic relatedness, and some of the geneticists are looking at that. So people probably do have a family history that they're not aware of. But where we get to with the people who don't have a family history, computations suggest that about 40% of the variability in the risk of getting ALS is genetic. So like most diseases, some of it's environmental, some of it's genetic. As it happens for ALS, the genetic predisposition is probably about 40%. The remaining risk is non-genetic or environmental, but that doesn't mean that genomics doesn't have a role to play, because many of the environmental exposures lead to epigenetic changes, which can be seen in DNA. Studies looking at prevalence and age of development of ALS in southern England came up with the idea that ALS does develop through a multi-step process, a bit like cancer mutations, likely 5 steps, but it could be 3, it could be 6 sort of thing. We also have emerging evidence that in both familial and sporadic ALS, there are risk genes. They're probably not causal. If you put them into a mouse, the mouse won't get MND, but they increase the risk. And we're also trying to get our head around modifying genes, a gene that isn't causal, but if you have the variant, it might make the disease go more quickly or maybe go more slowly, like those people who live for a long time. So the geneticists would consider sporadic ALS to be a complex disease, like hypertension or height or something. And we obviously need techniques to interrogate this. Such thing as a Mendelian disease. So this is finally the consortium. So we have now got involved in UQ and throughout Australia with large genome-wide studies. And clinicians' job really largely is to give well-characterised samples, to be very careful about the subtypes and the heterogeneity. We're really interested in looking, as we are already, at survival to try to figure out what links with long survival. And the SOLSHA consortium is Sporadic ALS Australia, Systems Genetics Consortium, run out of IMB by Naomi Ray, who's really the founder, and Anjali Henders, who is what we needed, which is a manager who's very strict about standard operating procedures and who helped put together the database, which is in Drupal. So we had those original patients who we had the GWAS done on, the whole exome sequencing done. We put them all into SOLSA all around Australia. Other partners have now put people into SOLSA, and then that gets put into Project Mine, which is based in the Netherlands, where they have a very strong interest in neurogenetics. So that's where we link up with the international consortium. And I've put this here, but just to say that only 2% of the risk, but it only explains about 2% of the risk of getting disease. But nevertheless, what I'm going to show you is the most recent paper. All right. So this has just come out, and this is a very large paper. It's something like the 6th GWAS study. And every time they sequence another couple of thousand patients and add it to the group before and do a meta-analysis. So the paper that's just come out has nearly 30,000 ALS patients, 122,000 controls, and 146 authors. So it's a very large paper, and it looks at linkage, and importantly, where I got very interested, it showed an association with the HLA region, which the previous 5 GWAS studies hadn't shown. And that's something that we're already starting to look at, because from all the sequencing data, it's possible to impute the HLA type. And this study showed a risk locus at this glutathione peroxidase SNP. So this is the results. I'm sure you've all seen these Manhattan plots. This is the C9orf72, or a link near the C9orf72. These are the various genes, which you can see along here. And this is the chromosome 6 HLA location. And this is the GPX3 locus that came up in this, as it had come up in previous loci. So we have contributed to this. It's, you know, very, very powerful study, but required a huge amount of collaboration. And I might say diplomacy to get all these people to work together. As part of the study, it showed something that Naomi and IMB are really particularly interested in, which is that ALS genetically associates with other diseases like Alzheimer's disease, Parkinson's disease, and progressive supranuclear palsy. And this is just a plot showing all of the different genes and how much the overlap is. To neurologists, we don't, we say, yeah, well, of course, if people get neuro disease, that, you know, they might have genetically, you know, predisposing factors in the brain. But this has really proven that. It's not just us speculating, it's proven that. And the other thing that this large study showed is by looking at expression data, it was possible to show that the genes that are implicated include glutaminergic neurons, which, as I mentioned, glutamate toxicity is one of the mechanisms put forward as the cause for ALS or MND. And it's actually, despite the zillions of dollars of clinical trials that have been done, the only drug that we've got that does anything at all to ameliorate ALS works on the glutamate pathway. Now, I won't go into this in any detail, but as I mentioned earlier, we've got the DNA. It's possible to look at epigenetics as well. And epigenetically, it also showed that the methylation profile is shared across various neurodegenerative disorders. But to do that, I've just listed there the huge number of cohorts that had to be diplomatically put together to do this large study. All right. Now, in Queensland, under at IMB, this locus has been pursued. I'm sorry if you can't read it, but I just lifted it from the paper, which we've now published. And it was a way of trying to tease out whether this GPX3 gene, which is near the locus, was the causative gene. And there's a huge amount of bioinformatics trying to model gene expression. This is this thing called HIDE, looking at expression pathways and protein interactions to try to provide evidence that the gene might be important. Stuff we got involved with, this is using some of the methylation and expression data that came from our patients. And then finally looking at models, and through our collaborators out at AIBN, there are now a number of lines of induced pluripotent stem cell motor neurons, and of course the zebrafish model out at the Brain Institute. And interestingly, all this evidence put this locus up the top, and knockout of the gene in the zebrafish led to a motor phenotype. So this has been published as, you know, strong evidence that this locus that emerged from the GWAS studies is probably important. Now, just mentioning some of the other stuff, because consortium to start with a complex disease and drill down to find causal genes. But for the clinicians in the audience, there is a way of doing it the other way too, where you can use this very complicated bioinformatics and sequencing to try to find the gene in a family. And I'm a small part of this consortium here, where a whole lot of bioinformaticians and sequencers have got together to do a lot of sequencing, particularly at the Garvan, doing this very fancy long read sequencing. As you know, when you do sequencing, you've got to kind of join the bits together till you figure out the sequence, and the long read sequencing helps. And so now with this project, we're getting informative families together, doing whole genome sequencing of all of the chromosomes of all of the members of the family, which is an enormous amount of data, figuring out ways of uploading that data and doing the bioinformatics with filters to try to find the gene. So conclusion, genetics is very exciting. There will be a role for clinicians. It's highly collaborative. The clinician is by no means going to be in charge of any of this. We have our own set role, but there is a need to work with statisticians and to learn their language. And we can put forward clinical questions. I was interested in one particular candidate gene, which is called the gene for speed. And they were able to say, yeah, we looked through these are the, this is from the whole exome sequencing. These are the genotypes, and it turned out that there was no suggestion of any linkage. So just to remind you, we've got the large consortium to try to sort out the complex disease, but we've also got a large, well-funded consortium to try to look at familial, more Mendelian disease. Now, this was what I just added this morning, is what I really learned about this. So the worst crime of all in this is low sample size. We really need large sample size. It's really important to learn about databases, and I've now been converted to this, I'd have to say, over the last decade. For those of you who don't know it, database is a very specific thing. It's not an Excel file. It's data that's linked together. That's the key part of a database. I have constructed a number of databases, and I'm a bit obsessional, but I'm the only person who's allowed to enter the data. I don't trust anyone else to do it. I think you do need to learn, I've mentioned R as the language, because even though you won't be doing the coding, when you go for a discussion about the results, you'll be just reading through pages of code. And I also to commend to you Bayesian statistics. I think as the computers get bigger and stronger, some of the stuff we've been doing involves the Bayesian statisticians, which I think would be a good thing for any young doctor to learn about Bayesian statistics. So thanks very much.
Excellent. Thank you, Pamela. And I love that last bit at the end there with regards to the Bayesian statistics. I remember working through that in pharmacy when we do pharmacokinetic drug modeling in a population. There is a question here. When considering both the genetic and environmental, are there any large retrospective cohort studies identifying the environmental factors in particular? Or are we still? So the answer is there have been many epidemiological studies, and they show certain obvious things. So people who are exposed to formaldehyde have an increased risk of MND. Formaldehyde is neurotoxic. And that's one of the things we've found, formaldehyde, even in our sporadic ALS patients. There are other, very large epidemiological studies that find things like working in a rural occupation is an increased risk. Interestingly, it wasn't just living in a rural occupation. You actually had to be on the farm doing the rural work and maybe some kind of toxins. And of course, there are well-known examples like the toxins in the Guam, the small country of Guam, where there are toxins in cycads. And again, part of our current work in the laboratory is looking for these toxic molecules in the blood, and we keep finding these
They're small molecules, so there may be evidence of toxicity. But drilling down further, part of what Naomi is doing with SALSA is including a very detailed epidemiological survey. It obviously won't have as many numbers as a huge epidemiological survey, but it will give very detailed epidata to go along with the genetic data. You've probably heard in the news that there's from time to, I think it was in the news recently, about whether the blue-green algae in ponds get into fish and you eat the fish and that's toxic. And again, the blue-green algae make a molecule like BMAA, which is the toxin in the island of Guam. So there's a lot to do, but I think the striking thing to me, and I don't know what, I can see David there, you know, you don't think of it as a common disease, but in fact at Royal Brisbane, we've got 800 MND patients on our list. We've only got 500 MS patients. So even it's you know, it's not a rare disease. Thanks.
Certainly. Thank you. Another question here. Some clinicians undertake Master of Philosophy or other postgrad coursework masters to gain statistical database skills. Do you have any opinion on this type of training and like the, I guess?
So I think it depends on your kind of interest. Some clinicians want to actually do the bioinformatics and do the statisticians and have a career in public health or epidemiology. I think you probably should do the training. In terms of being a good collaborator and consumer, I think you can learn a certain amount. Just, you know, I mean, with the Bayesian statistics, we just worked with a professor at QUT, and we had virtually personal training until we could understand how the Bayesian statistics works. I think in terms of, you know, learning coding, I mean, they're teaching kids coding in primary school these days. So I think it, you know, learning coding, learning R is something you can do through YouTube, basically. And so I think the answer is what your preferred destination might be.
One of the very common things I do get told as a PhD student is to learn coding just so that you can automate some of the analysis that you're inevitably going to do. So I think that the value in that is definitely not small. Just as an additional question, where do you think most clinicians' understanding of complex trait genetics is at? And do we need a greater awareness? And if so, how can that be encouraged?
I mean, I think we do. I think it'll be kind of incremental. I mean, maybe at medical school more could be made of it, but I think it's, you know, even if you think you're interested in Mendelian genetics and, you know, a disease like Charcot-Marie-Tooth disease, it's blindingly obvious how diverse the phenotype is, that there have to be other things going on. And I think it's going to be sort of gradual and incremental, yeah. But putting together, you know, genes and environment in the one place, I think is one of the strengths that SALSA will grow into eventually. And, you know, to some extent, the Project Mine is doing that too, although it's more difficult to kind of correlate all of the different countries. And, you know, clearly, you know, we didn't like it when Angela came out and said, you have to do it this way, you have to do it this way, but it's the only way. We have to have standard operating procedures. We can't just do it the way we want to do it. We have to, you know, properly participate in a database, which was a bit new to us.
The researchers doing meta-analyses in the future will very much thank you for that.
Yes, yeah. No, I mean, I think it holds true, doesn't it, for all studies. There ought to be a kind of minimum data set for your MS or your hypertension or whatever, so the meta-analysis can happen, absolutely, yeah.
Thank you again for an excellent talk. It was quite enlightening, really.